Hello everyone, welcome to this ultimate guide for access to HE. Today we'll be talking about barriers to infection as part of the infectious disease unit. The slide shows the structure of the human immune system, and obviously the immune system is at the top, and you can see that the immune system is split into two branches. Now the two branches, although can be identified as separate, they work together to protect us from all the different pathogens that are swarming around us in the atmosphere and everything that we touch, etc. The system on the right is called the non-specific system or the innate system. It's a little bit more tricky to remember that, but if you can, it's a little bit more scientific. Either will do. And the strands of the non-specific system that I've chosen really to talk about are the top three. There are other things as well, but we just don't have enough time and enough space to talk about everything, and you'll have plenty to write about. So the first thing we're going to talk about is barriers to infection, which is the presentation today. It's part of the non-specific or innate system. Now, non-specific systems or the non-specific system means that it's the part of the immune system that doesn't really respond to specific pathogens. It just knows that this thing is foreign and it shouldn't be there and we're going to do something about it. It doesn't identify that this is the flu virus or the adenovirus or anything else, any other pathogen. So that's what we mean by non-specific. It doesn't really identify what it is, it just knows it shouldn't be there and it responds. One of the beauties of this system is that it's extremely fast. So if something does manage to break your mucous membranes or get inside your cells, or inside your body, should I say, then the non-specific system will work very, very quickly. The other strands of the specific immune system are the inflammatory response, which most of us, if not all of us, would have felt at some time when your tissue gets red, swollen, and hot. And phagocytosis, which actually you know more about than you think, particularly if you can remember your cell biology unit. Now, the specific or adaptive system kicks into place once really a pathogen is identified. And these two systems work in hand, like I've said, to destroy the pathogen that has been identified. But this specific system, i.e. the humoral response and the cell-mediated response, it takes a little bit of time to get into full action. So we felt this before. When you come down with sort of flu-like symptoms, it takes really sort of a week to start actually becoming better and fighting it. And that's because it takes time for the body to recognize what it's fighting against. And it can't go around with all the T cells and B cells, the white blood cells that are trained killers for specific pathogens, because there are so many. If we had so many white blood cells in our body, we'd be walking around like Marshmallow Man. It'd be, we'd be absolutely filled with white blood cells. So what has to happen is that once we've identified a threat, then we do something specific about it, and that's when we release these sort of assassins of the white blood cells. These white blood cells over here are generic killers. They will just basically munch up anything that it does find, but it will also activate certain specific killers, assassins, that are trained to nuke that particular virus. Okay, or pathogen, should I say. And that's an overview. Today we're going to talk about barriers to infection. It's our first port of call. So, if you think of your body as a castle, all right, some are stronger than others, what obstacles would you put in place to stop invaders getting in? Well, classically, a castle has walls, and it has, a lot of them have moats, and they're barriers for an invading army. I can tell I'm having fun. From getting into and over the walls. Now, these are things that are put in place to slow down and protect against invaders from getting into the castle. And our bodies are very much like that. We have physical barriers and we have chemical barriers. They're split into two. Now, I would like you to learn about both of them. So, for example, in our castle, the physical barriers would be the wall, okay, and the chemical barriers might be, let's think about that moat filled with acid. I'm just going to change it a little bit, and if you step into that acid, your face will melt off, and that would be a chemical barrier. It would basically stop that invader from getting to the wall, essentially, and so that's what we mean by a chemical barrier. So your bodies have both physical and chemical barriers within the body. Now, lots of them are common sense, and we'll go into them a little bit more detail. So, a question which is a rhetorical question, whereabouts in the human body do we need barriers? The simple answer is that we need them wherever the outside world meets our bodies. So any mucous membrane, any membrane that is a barrier between the outside world. So our skin is the obvious one, you know. It has certain physical and chemical properties that help us to defend against all the millions of pathogens that are surrounding us. The eyes, the digestive tract, the respiratory tract, and the ears. So you have to sort of imagine that all along these two tracks here is actually the outside world. It's a gross thing to think about, but if you take a deep breath in, the space that goes down your trachea and your bronchioles and fills the lungs is actually the outside world. There's no barrier between the atmosphere of your when you breathe in to filling the lungs, and so there has to be barriers and physical and chemical properties of that lining of the tubes that can protect against pathogens. And it's the same goes the digestive tract. What a beautiful image this is. When you eat something, i.e. your mouth, it goes down the esophagus and into the stomach and small intestine, large intestine, and it comes out of your anus. So actually the tube from your mouth to your anus is in fact could be classed as the outside world. And so all along that tube is the outside world. So all along that tube, we need physical and chemical barriers that are going to protect us from the pathogens that are around us in our food, in our water, etc., etc. Your task is to really prove that you've done some research about it and that you have a solid understanding. You're not going to be able to talk about all of them in the assignment. Have a look at, have a research about the different bits of barrier that we have on our body systems. The different parts may well take your fancy. You might want to learn about how the eyes protect, or the respiratory system protects. It's completely up to you. You have a free reign of choice. What I'm going to do is set the scene as to what the standard is that we're looking for in terms of the knowledge and how you portray it in your assignment, really. So this is just a few examples of types of barriers in terms of physical and chemical. Like I said, the skin has both physical and chemical. You've got stomach acid as chemical, saliva, tears, any kind of fluid and I suppose chemical that is released by the body that helps protect. And then the physical barriers, you've got hairs, nostril hairs, you've got cilia of the sort of pipes which we'll go into, and the mucous membranes. I thought this was useful because it's a summary of the skin. Now, I'm not going to talk in detail about the skin, but the skin has sort of some common sense properties because, you know, we have skin, we can see it, we can touch it, we can relate to it in terms of how it protects us. If you want to go into this, by all means do so. I'm not going to go into more detail than what you can see on the screen, but it's got a waterproof layer. It doesn't let everything in. It's made of keratin, which is a tough structural protein. Interesting that it sheds, a bit like a snake skin, really, I suppose, but not in one. Anything that attaches to the skin will not stay there for long because we shed it. We replace it every single day and every single week of our lives. We're able to clot the blood. We're able to clot the skin with blood clots and therefore help to repair. In terms of chemicals, we've got an acid mantle, so it's slightly acidic, is the skin usually. And any pathogens have to compete with natural flora on our bodies. You know, there's almost as many bacteria as there is R cells on our body, which will completely freak out a lot of of the germaphobes. But it's true. You know, you're surrounded, you are being lived on by lots of different bacteria, and actually that is competing with the nasty pathogens for space, for nutrients that you're giving off. And I'll let you read the other two. I'm not going to sort of flog a dead horse. So what we're going to do, or what I'm going to do, is go through what I would do if I were to do this task. So if I'm looking really at the structure and function of a system, and looking at the physical and chemical barriers of that system, so that I can really get some high quality information in only, let's say, 200, 250 words, maybe a couple of diagrams. But it's about the quality of work rather than the quantity, and it's really about trying to get in your understanding supported by things that you've read in a way that's your own. You have to make it your own. So I'm going to go through the approach that I would take, and hopefully it's useful, because you really need a kind of resilience to when you go into doing this kind of work at a really super high level, because you may well find research, you might find words, and you might just, some people assume that the reader knows what the words mean, but it's really up to you to show what you know or what you mean. So anyway, we're going to look at the trachea, the respiratory system, so the windpipe essentially, and that's what we're going to have a look at today in detail. Now this is a cross section of the windpipe, so this is like sort of one ring looking down, so if the camera's sort of looking this way, and you can sort of see this is the barrier, I suppose the physical barrier between the outside world and the inside world. All right, so this barrier, this mucous membrane, has lots of properties to protect us from harm when we breathe in, because we're breathing in pathogens. You don't have to walk past somebody or walk in a classroom. There's going to be bacteria and virus and things that are floating around, and we have to be able to deal with them. So let's have a look at how we do that. So in my research, a good book at the bottom there is by Albert et al. And friends. It's the molecular biology of the cell. And we can see, this is where I first started my research, is that the three types of specialized cell that line the respiratory epithelium, which is the lining of the airways. So this is a really good start, because it tells me the three types of cell. You've got goblet cells, ciliated cells, and endocrine cells, which basically mean hormonal cells, or cells that create hormones. So using any resource, create a mind map on a piece of flip chart paper relating to the structure and function of a goblet cell, which is what I've essentially done. I've gone through the process that I would have done as a student, and I go through that for the goblet and ciliated cells, looking at what the physical barriers and the chemical barriers are, and it gives you a bit of a clue at this top of the picture of where I'm going with this. But let's go for what we would have found. You can by all means go for those, which is where I found a lot of information. So here we can see this is a real sort of microscopic view. You can see that the goblet cells look like a sort of goblet, a wine goblet. I'll put a little base on there. You might be drinking some Prosecco at home. It's like a goblet. And then you've got the ciliated cells next door. You can see these. You can see that actually the goblet cells, they do have some cilia on them as well, but you'd see that these have really fine hairs. Okay, so that gives me an impression of that they're working together. So let's have a look. Here's a passage from Marieb, which is my favourite textbook in terms of human physiology. Marieb. Its cilia continually propel debris-laden mucus towards the pharynx. So we know what cilia are. They're like fine hairs that beat in the same direction. So if anything got trapped in the mucus, anything that got trapped in the lining of the windpipe, the trachea, then these cilia would propel the mucus, or the debris-laden mucus, towards the pharynx, so towards the back of the throat. It's a good start. So I also then wanted to look at the mucus. So I found in this journal over here that particles are normally trapped in the luminal mucus layer. So this mucus layer, this green stuff, as you can see on the diagram at the top, the pathogens are going to be trapped in the mucus. So that leads me to believe that the mucus is going to be sticky. Okay, so I'm picking out little bits of information from different sources, so I can possibly link that to the previous slide in that about the cilia, and I'm just sort of piecing pieces of the puzzle together. So here's where I came up against another respiratory journal. Basically, this was entitled Responsive and Adaptable Frontline Defenders. So depending upon species and preparation, goblet cells discharge mucus in response to a wide variety of stimuli, including irritant gases, inflammatory mediators, and the environment. So basically, what that means is that if you, let's take smoking for example, if you smoke or if you're in a smoky building, then goblet cells will discharge in response to that stimuli. So if you're really in a smoky environment, then mucus will start to be produced and released. So new information demonstrates that goblet cells can discharge vast quantities of mucus in fractions of a second, a property integral to airway defense against acute insult. So basically, in medical terms, if it's been damaged or if it's compromised by smoke or toxins or anything like that, the goblet cells will just shed lots of mucus to try and stick whatever's coming down the airway. So I'm thinking, well, okay, so that could be a physical barrier in terms of the mucus. It sticks things, it stops things from moving. So I had a little bit of a look into what is mucus, what's in it, what can that do. Now here we've got a bit of trapping, so we've already said that, but I also picked up that it's got something called lysozyme in it, which is a protein, and that got my mind thinking, okay, what is lysozyme and why would it be made of protein? So I did a search on lysozyme, and lysozyme is an enzyme that speeds up chemical reactions, so that's what an enzyme does, that breaks down the bonds in the cell membranes of bacteria. This reduces chances of infection. So I had a real sort of penny drop moment there when I sort of searched about lysozyme and I could link those two sources together. So it doesn't mean that you're going to be writing sentence after sentence, but you can quote the two, which basically means from the last quote that mucus contains lysozyme, which according to this source, and I'll leave it up to you to find which source is an enzyme that breaks down the bonds in the cell membrane and reducing the chance of infection. You've got beautiful information that you've researched through. It's like a puzzle that you've unlocked the different bits and bobs. And here we've got a picture of a goblet cell. You can see it's got microvilli underneath. It has a nucleus at the base. Okay, lots of Golgi apparatus in the middle section and lots of vesicles, bubbles stored, ready to be released into the lumen of the air windpipe. So the windpipe's over here. Now this is the space of the windpipe. So as soon as this starts filling up with smoke or toxins, these vesicles will just move to the top. Guess which process the vesicles release the mucus from, hopefully you guessed it, exocytosis. Merges with the membrane, and this mucus spills out into the airway. Now, a key annotation at the bottom there, which I've circled, it says, notice the secretory vesicles and well-developed rough endoplasmic reticulum and Golgi apparatus or apparatus. So they're well-developed. Why would they have well-developed endoplasmic reticulum and Golgi apparatus? Why? Well, the answer would be simply that they are responsible for the packaging of mucus and lysozyme into these vesicles. So you can see it's kind of a factory. Here is the brains, here is the code in the nucleus, sorry about my writing, to create lysozyme. The code then gets replicated and sent to the ribosomes to be made into lysozyme, and then to the Golgi bodies to be packaged into these vesicles ready to be secreted. So you can really talk about the structure of a goblet cell, and that's really how it's so responsive to insult, like we said on the last slide. As soon as this starts filling up with smoke or toxins or whatever, these vesicles are ready to go, and your lining of the airways can just fill with mucus and sticky stuff to try and trap the pathogens or protect the lining of the airways from harm. So another question that cropped up in my mind is that the goblet cells have microvilli. They have microvilli, which are a lot smaller than cilia. They're microvilli. So I'm just wondering, in my head, come across this research, why do goblet cells have microvilli? Can you think? Well, microvilli is a common sort of principle in terms of human physiology. The principle of increasing surface area. So if you had a flat surface, let's say it measures 5 centimeters, that's how much stuff can travel across this membrane, because it's a flat surface. You can only sort of, the rate of diffusion, or whatever method it's coming across, can only transport stuff at a certain rate. But if you make certain folds in the membrane, you may well extend this line to, let's say, 10 centimeters. You've doubled the surface area in contact with the outside world. So you may well be able to absorb or transfer whatever twice, 3 times, quadruple the amount of material that you would have done if you had a flat surface. Okay. Now this is where microvilli come in, because the microvilli are not just bumps in the membrane, they're actually mini bumps on the folds of the membrane. So having microvilli may well have increased the surface area to 30 centimeters. So where you had a 5 centimeter straight line, you've now got a 30 centimeter jagged line, which means that you can basically absorb, soak up, give off far more than you would do with a straight line. And that's the principle of surface area. So why would that be important for a goblet cell? Well, hopefully you've guessed it. The more microvilli, the more mucus can be released in a faster time, and that helps the goblet cell to release its mucus in response to insult from toxins, smoke, blah blah blah. So here's a quote: goblet cells lack cilia and have occasional microvilli which increase the surface area for secretion. Boom. Quote. I would paraphrase that into my own sentence to make my understanding clear, and it's been informed by expert sources, but it's me that's written it, and it's me that's found the information and pieced it together. So this is a summary of the goblet cell. It's got Well-developed rough endoplasmic reticulum and Golgi bodies. Why? For the production of mucus. It's got microvilli to increase the speed or the rate of transfer of exocytosis of the mucus. So have a go. Have a go at writing how a goblet cell of the respiratory pathway is suitable for as being a barrier to infection. Now at this point you may well need to pause the video and have a think, and I'll do a little bit of a summary off the top of my head, but in your assignment you're trying to evaluate the effectiveness of the non-specific system. So how good is it at protecting us from harm? So in terms of the goblet cells, you've got microvilli, which increase surface area. You've got well-developed endoplasmic reticulum and Golgi bodies, which make sure that you can produce the mucus in time. And the mucus itself has a protein in it, and a lysozyme protein in it, which breaks down the barriers of the bacteria and also provides a sticky surface for them to stick on. So you've got kind of 4 or 5 points there that you can use expert information to support what you say. Now, the other type of cell that was talked about right in the very start was a ciliated cell. Now, a ciliated cell has, I suppose, like hair-like structures that stick out of it. They're very, very small, but here we can say the mucosa has the same goblet cell containing pseudostratified epithelium that occurs throughout most of the respiratory tract. Its cilia continually propel debris-laden mucus towards the pharynx. Okay, so it's really a joint approach. The goblet cell creates mucus. The ciliated cells propel that mucus towards the pharynx. So they really work closely together, one to try and trap it and to eliminate it through the lysozyme. And the ciliated cells beat their cilia to the back of the throat where we can spit it out. So it doesn't get down into the lungs. It's very, very difficult to deal with pathogens that get into the lungs. So a little bit of research was done about cilia. So what are they? It tells you a little bit about the structure of the cilium at the top here. And it says the primary function of cilia is to move fluid over the surface of a cell or to propel single cells through a fluid, which is some pathogens are able to do that, like protozoa. On the epithelial cells lining the human respiratory tract, huge numbers of cilia, more than 1 billion per square centimetre. What? 1 billion per square centimetre? That's insane. Sweep layers of mucus containing trapped dust particles and dead cells up towards the throat to be swallowed and eventually eliminated from the body. That's a beautiful quote which sort of summarizes the function of cilia and exactly how many there are. I didn't actually know that there was that many until I read that. And it keeps going until it gets all the mucus towards the back of the throat. Here is a bit of a diagram as to how a cilia contracts. It's basically a one-way system, so power stroke that way and it goes floppy and retreats that way and then power strokes again. So it basically makes sure that things go in one way rather than another. Suppose like rowing a boat. You don't want to row both ways because you'd never get anywhere. You pull one way, lift it out of the water, reset, and then back in the water and sort of do a power stroke again. You can click on the link, follow the link, and there's some YouTube footage of how to do it or how it's actually done. Another few quotes that I found in my research: mucociliary clearance is a major function of the airway epithelium. Basically, it's clearing the airways. Basically, airway mucus on the activity of the cilia. The former, in turn, is dependent mainly on the quality and quantity of mucus glycoproteins or mucins, which are produced by 2 different cell types. namely goblet cells or epithelium. So really we've come across a couple of new terms there, glycoproteins and mucins. So I wanted to sort of look into that a little bit more. But I'm going to leave that with you for the time being. And this is my sort of diagram of sort of the escalator effect. So you can see the goblet cells are in sort of this lighter green. They spew out the mucus into the airway, and the ciliated cells move the stuff up towards the back of the throat. And that's sometimes called the escalator effect, the mucociliary escalator. Okay, so cilia. The final question really is about why the cells are tightly packed, because on the original quote it said the pseudostratified epithelium cells are tightly packed. So I'm going to ask myself the question, why are the cells that surround the airways really tight, really squeezed together? And the answer is relatively obvious. You don't want gaps between the cells because that'll allow pathogens to get in and cause problems. But how is that achieved? Why are the cells really tightly packed? Okay, a few definitions which you can pause before we move on. But really what happens is we have something called tight junctions between cells. And what that is these things here are proteins that span the membranes of neighboring cells. So this is a cell, this is a cell down here, two cells together, and it's the join of the two cells using a protein so that this pathogen here, this bug, can't get between the joins of the cells. So I've written it there, bacteria, dust and other unwanted substances cannot lodge between the goblet cells, gain access to the bloodstream or start replicating and causing infection and disease. So what I would try and do is I'd try and find a quote or some kind of information from a relatively reliable source that supports the tight junctions. Okay, so it's now your turn. I've put 700 words there, but I think that's going to be too many for your assignment, but at least it'll give you a guide as to what to start with. 2 diagrams, 2 references as a minimum, but if you weren't writing 700 words, I'd expect possibly up to about 10 citations and references. And what you're looking to do is explain how or evaluate the chemical and physical barriers to infection of the respiratory system. Okay. So just as a summary, I know I'm going back over all ground, but you've got goblet cells and ciliated cells basically working together to form a sort of escalator effect on the trachea. The goblet cells produce mucus, and they do that because they've got well-developed endoplasmic reticulum and Golgi bodies, which put them in vesicles to store ready to be released. The mucus is very sticky, and it also contains lysozyme, which breaks down bacterial walls. You've got microvilli, which increase surface area and basically mean that it's a faster rate of exchange and release of the mucus. Corresponding to that, you've got ciliated cells, very thick with little hair-like structures which work together to propel the bacteria that's stuck in the mucus towards the back of the throat for either spitting out or for being swallowed. You've also got the physical barrier of the cells being joined together very tightly by tight junctions, and they're proteins that join cells together and do not provide any entranceway for pathogens or dust or anything like that into the bloodstream. So you've got all those things to write about, supported by quality information and sources. You've got plenty of things to get your teeth into, so get researching, get pen to paper or start typing and see what you can come up with in terms of a quality bit of writing. I hope this is been an interesting little video screencast. Like I said at the start, you need resilience in your study. So you may well find words that you don't understand. You may well find words that lead you to a source that is far, 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 far too detailed and academic. But don't be afraid. Find a way around it. If you find a dead end, just make your way around and go around. You may well have to go sideways before you can go forward. But the key to being really successful is do not accept not understanding. Don't give it up. If you really struggle, come and see me and we can work it out together. But if you get to a particular point where you think, I know this needs to be in there, don't just say I can't be bothered, or don't just throw in a sentence that you don't understand. If you don't understand it, there's a good chance that I'm going to pick that up when I come to market. So make sure that you understand it and that you show that you understand it in your work.